Okay, so today we're going to talk about the cell cycle. So here we go. So the cell cycle is the process in which cells divide and is made up of interphase and the remainder of the cell division process. The cell spends approximately 90% of its time in interphase while it's dividing, as you can see here. So about 90% of the time it's spent in interphase. And then the rest of the 10%, it's in the mitotic phase, which includes metaphase, prophase, anaphase, and all of that good stuff. It spends most of its time in interphase because DNA replication and other important processes are occurring during that time. And both interphase and mitosis have stages that help them progress. Interphase and checkpoints. So there are 3 phases or checkpoints during interphase. So here are the 3 phases. So there's G1, S, G2, and mitosis. And so there are 3 phases in interphase, G1, S, and G2. And then M is just the process that occurs during mitosis. And so there are also checkpoints. So there's the G1 checkpoint, the G2 checkpoint, and then the M checkpoint. The G1 phase is known as the first gap, S is known as synthesis, and G2 is the second gap. And then, like I said, there is also an M phase, which represents mitosis and cell division. So during all of these phases, the cell grows by producing proteins and cytoplasmic organelles. During G1, the cell grows. So during this part, the cell just grows. Next, the synthesis phase is where the chromosomes are copied. So this is where the chromosomes are copied. In the G2 phase, the cell prepares to be divided. So here, it's preparing to be divided in the G2 phase because in mitosis phase, of course, the cell is going to be divided. These phases would not be able to continue without the checkpoints that are giving the cell signals to continue on to the next phase. So, for example, if the cell is at the G1 checkpoint, it would not be able to continue on to the G2 checkpoint until it got a signal saying that it was okay for it to continue on. There is also a stage called the G0 phase, which surprisingly is where most of the cells in the human body are. If no go-ahead signal is given to the cell, then the cell enters the G0 phase and won't divide any further. For example, in our body, mature nerve cells and muscle cells never divide. So next we're going to talk about internal and external signals. So these internal and external signals are things that alter the way the cells divide. One example of an internal signal would be during the M phase, or during mitosis. So during anaphase and mitosis, the centrochromatids are not able to separate until the chromosomes are aligned on the metaphase plate. This picture here is actually an example of an external signal because here you can see there's just skin tissue, and then it is added to these little tubes, and one is given a nutrient called PDGF, and one is not given PDGF. And so the one that has does not have PDGF is not going to divide because the cells need that nutrient in order to divide. And then the one without PDGF right here will divide because the cells have that nutrient that's needed in order for the cells to divide. So this would be an example of an external signal, because there is something outside of the cell causing it to not be able to divide. And then the example that I gave of the sister chromatids not being able to separate until the chromosomes are aligned would be an example of an internal signal because it happens inside of the cell. So next is cyclins and cyclin-dependent kinases. So the molecules that regulate the cell cycle are made of proteins called kinases and cyclins. Kinases give go-ahead signals at the G1 and G2 checkpoints. Most of the kinases that drive the cell cycle are present at a constant concentration to be active. These types of kinases must be attached to a cyclin, which gets its name from its changing concentration. So the kinases have a constant concentration until when they are inactive, and in order to become active, they have to attach to a cyclin. And so the cyclins get their name from their changing concentrations. And so the cyclin level rises during the S and G2 phase. So as you guys can see, the cyclin concentration right here, right when it gets to mitosis, it drastically decreases. And then when it gets to that S, it begins to decrease. And then when it gets to the M, it decreases. And that's the pattern that it keeps. So it rises during the S and G2 phase, and then it decreases during the mitotic phase. And then MPF is just reliant on the cyclin concentration, so it corresponds with that. So as you guys can see, as the cyclin concentration increases, the MPF activity is also going to increase. And so it just follows the same increase as it goes along. So finally, cancer cells. So cancer cells are really crazy because they do not follow the same routine as most cells do and do not heed the normal signals. So if a normal signal would pick up on the go-ahead signal during the cell cycle, the cancer cell is not going to pick up on that same signal. Unlike other signals, cancer cells will continue to grow uncontrollably without considering internal and external signals. When cancer cells do stop dividing, they do so at random points. So usually, during the cell cycle, the cells will stop growing at those certain checkpoints that I was talking about. At the G1 checkpoint and the G2 checkpoint, they'll stop growing at those checkpoints until they get the go-ahead signals. But cancer cells, they'll skip right through those checkpoints and just stop growing at random places instead of stopping at those checkpoints. These cancer cells, when cancer cells do stop dividing, they do so at random points. These cancer cells can all gather in one place and cause tumors. As you guys can see in the picture here, here's a tumor, and then as they pass through these checkpoints, they can all gather together. And if they do pass destruction, that makes it much easier for them to gather together as well. Some tumors are harmless, like benign tumors. Those, you can get them removed, they're not a big deal. But then there are I'm not really sure how to pronounce this, Magellent, I think, tumors. And these are very harmless and can cause harm to the organs. The other really bad thing about cancer cells is that they can break off of these tumors and actually go through the blood vessels. As you guys can see here, they can go through the blood vessels and move to other parts of the body, which is really bad because you never want cancer in more than one place of your body. And so that's called a metastatic tumor. These tumors, and they can spread to other areas, and that's called metastatic tumors.